being an academic, physician, I'm never satisfied with the status quo, right? Because there's always patients that I treat that fail to benefit from the standard FDA-approved therapies right now, and I think it's important to create new options for those patients.
This is the James Cancer-Free World podcast. I'm Steve Wartenberg, and my guest is Dr. Richard Wu. Richard is a medical oncologist who specializes in skin cancer and melanomas and the early development of new drugs to treat these very common forms of cancer. In a previous episode, episode 122, Richard filled us in on TIL, T-I-L, which is tumor infiltrating therapy, a new treatment in which a patient's T cells are removed and grown in a lab and then returned to the patient's body where they are better able to detect and target cancer cells. Richard mentioned a new drug being developed that would improve the TIL process, and that's what will fill us in on today, as well as the future of TIL and what it could mean for fighting cancer. Welcome back to the podcast, Richard.
Yeah, thank you for the invitation, Steve.
Great to have you back, and I'm excited to learn more because the last time you talked, you briefly filled us in on a new drug that was going to make the TIL process even more effective. So I thought we would start. I gave a real quick thumbnail sketch of what TIL is, but expand a little. What is TIL, and sort of where are you in the process of clinical trials and treating people with it?
Yeah, so TIL stands for the tumor infiltrating lymphocytes. It's a different approach of the cancer immunotherapy. So as you know, right now the currently FDA-approved immunotherapy essentially, they give you a antibody-based drug that sort of seems, that intends to unblock the immune system so they can recognize and kill the cancer. The TIL works a bit differently. So what happened is that within each patient's tumor, is often infiltrated with these cancer-fighting immune cells called the T cells, and they can recognize quite a bit of unique characteristics that can be present in each patient's tumor. It's a very personalized kind of therapy. So the idea of TIL is to isolate these T cells from each patient's unique tumors, and expand them to billions into cells in a laboratory in a process called a rapid expansion protocol, and then reinfuse back into the patients that are lymphodepleting, meaning that they have received chemotherapies to kind of deplete out their existing immune system to make space for the new one, and also the IL-2, which is the T cell growth factor that will expand out these TIL once they're infused back into the patients. It's a very personalized, in my view, a very much more personalized form of immunotherapy, and I think that may explain of its unique sort of mechanism of action and potential efficacy. Yeah.
So if these T cells are already in the cancer site, but are they not working? They're not recognizing the cancer cells?
So what happened is that within each patient's tumor, it's a very hostile environment for these T cells. And these T cells actually, they're doing what they're supposed to do. They're infiltrating the tumor. But once they get there, because of the hostile, the tumor microenvironment, that's the term that we use to describe it, these T cells, they're not able to properly, differentiate and become the potent cancer-fighting killers that they're destined to be.
Now, is that because the cancer cells somehow put up resistance? They do some sort of resistance to stop the T cells from doing their job.
Yeah, my previous research have shown that that in fact the tumors make a type of a signal molecule called we call it cytokines called the TGF-beta that actually arrests the T cells from properly differentiating into the potent killer. So as you know that the T cells had to become from the they had to go from like the younger sort of like a less experienced T cell to become the more experienced T cell. It's kind of like going from like novice to a veteran, right? But the problem is that once the TIL infiltrate into the tumor, they're blocked in that process of becoming an experienced killer. So that's why that's why despite seeing all those T cells inside the patient's tumor, when they're diagnosed with a melanoma, we don't see a tumor spontaneously disappearing for that process. Yeah.
Oh, and I'm glad you mentioned that in a melanoma, because I think what you mostly study and treat is people with melanoma with the TIL process.
Yeah.
but I think we will get to it. There could be other applications to different types of cancer, but for now, we're talking about melanomas, which I checked, and there's 97,000 cases a year of melanoma and 7,000 deaths. So this is much needed advancement in therapy.
Yeah. Yeah. So essentially, with the TIL process, because because these TIL they're not differentiating normally in the tumor microenvironment. So the idea is that if we isolate them out and grow them in a laboratory and push them to become the more differentiated, potent cancer-fighting T cells, then that can potentially make them more effective. And that's really what I think is the one of the major principles underlying the efficacy of TIL. Yeah.
So you're multiplying by mega thousands or millions the number
billions,
of billions, the number of T cells. But are you changing them in any way? And is that where this new drug comes in, or there was already a way to change them to make them more effective, and now this drug will add to that?
Yes. So actually, in the manufacturing process for the TIL, we're actually changing the characteristics of its TIL. So the one aspect of it is to push them to differentiate to become the potent anti-cancer T cell that they're destined to be. But they weren't able to do that within the tumor, but now they're able to do that in the laboratory.
You make them smarter, stronger.
Yes, and to kind of push them through the normal T cell differentiation pathway to become the potent killers. And also we're also increasing the amount of tumor killing molecules inside these T cells. So my previous research also has shown that during this manufacturing process for the TIL, the T cells started to increase the expression of molecules that's typically associated with tumor cell killing, such as granzyme B and perforin. So you can think of a granzyme as the bullet, and the perforin is like a barrel of a gun. So
say those words again.
So essentially, the T cell, the TILs, they increase the expression of these two molecules that are essential for tumor cell killing.
Oh, okay.
One of them is called granzyme B, and the other one is called perforin. So you can think of it as a gun and barrel. So the perforin basically punches a hole on the tumor cell's membrane, whereas the granzyme B kind of goes through that hole and actually initiate the cell death process in the cancer cells. So my previous research has shown that when TILs are grown in the laboratory, they start to kind of express these molecules that are essential for tumor killing. Yeah, this is how this new drug works.
Oh, okay. So let's introduce this new drug. Give me a little bit of the history of it and where it is now, and what is it called?
Yeah. So this TIL therapy actually has been in development for almost 2 decades now. It was initially developed by Dr. Steve Rosenberg, who is a very prominent physician scientist at the National Cancer Institute, and he's a surgical oncologist by training. And he published a very important paper in 2005. For the first time showing the feasibility of growing T cells from patients' tumor and treating them as a form of this TIL therapy. And he has published some really impressive papers with patient images. One of the images that he likes to show in a conference is a patient with a very bulky tumor on his neck who experienced a complete disappearance of a tumor after the TIL therapy. Wow. Okay. And since that, there has been a couple of promising trials demonstrating the efficacy of TIL. So this company called Iovance, they have they sort of grew out of that research effort in an attempt to try to make the TILs grow the TIL on a larger scale to make it more to make it into a potential commercial drugs. And as far as I know, last year they were going through last year the FDA was asking them to submit some additional information about how to ensure the potency of each patient's TIL product. They had to develop some sort of test to be able to do that, and they were able to accomplish that goal early this year. So they have resubmitted the application back to the FDA for approval, and so the FDA has until November 25th to act on it. So my anticipation is that approval could either come before the deadline of November 25th or close to it. But it's a very exciting time now.
Well, we're going to take a quick break, and then we're going to talk a little bit more about exactly how this drug works.
You didn't choose cancer, but you can choose where to treat it. And when you choose the James at Ohio State,
you're picking a team of experts who understand there is no routine cancer.
You're opting for care from a highly specialized team dedicated to treating one type of cancer, yours.
A team that studies the unique makeup of your disease to develop a personalized treatment plan.
You're choosing our region's only comprehensive cancer center designated by the National Cancer Institute.
Where more than 1,700 scientists are working on new treatments and new hope for every form of cancer.
At the James, you're making the choice to have the most advanced treatments, many of which were developed right here.
And you're choosing access to the James world-class clinical trials, dedicated support services, and an unmatched survivorship program to support your life after cancer treatment.
You didn't choose cancer, but the choice of where to treat it is clear.
We're back with Dr. Richard Wu, and we're talking about a new drug that's going to improve the TIL process. And he gave us a great overview, but I'm always fascinated by the process of how this works. So kind of walk us through when you have a patient in your clinic, how you remove that patient's T cells, what exactly is done to them with this new drug, and then how they're put back into the patient, and then what happens.
Yes. So the TIL process it's a very involved process. So initially, the patients would have the certain size of tumor that can be safely surgically resected. And for example, like let's say if a piece of tumor is in the lungs or in the liver, they have to be of certain size, because that's very important to ensure the success of a TIL. If a tumor is too small, the chance of growing TIL is much less.
There won't be enough T cells in the tumor to remove to grow.
Yeah.
And then another thing you said, these are metastatic melanomas that have gone to the lung and to other parts.
Yeah, other organs, yeah.
Okay, so it's still, you're treating melanoma, but it's just gone to another part of the body.
Yes, so essentially we're taking a piece of one of those tumor lesions.
Okay.
And so after these tumor lesions are removed by our surgeons, they will then get sent to a facility. This facility is actually pretty fancy. They have like bioreactors, and so essentially these tumor fragments are cut into like little pieces, and they are grown with a cytokine called IL-2 into these bioreactors, where they can expand to like billions of cells.
Wow, billions. Okay,
yeah,
billions of cells are still a little tiny drop.
Yeah. Yeah, and actually studies have also shown that the amount of T cell that's infused into patients is very critical to success. There's a threshold that below which that efficacy can be much more reduced if there's not enough of those TILs.
So you need billions, not hundreds of millions.
Yeah.
Okay.
And so usually this process takes somewhere between like 4 to 6 weeks. Again, the speed of growth is very different, depending on each patient's biological makeup is. And I think my experience, the younger patients tend to have TILs that can grow like a little bit faster than older patients. Yeah.
Oh, interesting.
Yeah, again, every patient can be different in terms of speed of growing these TILs. So then after this TIL product is ready, so the commercial product is called lifileucel.
Say that again.
So this commercial product is called lifileucel.
That's the new drug.
Yeah.
Okay.
Yeah. So once this product is ready, the patient will then be arranged to be admitted to the hospital, where they will undergo roughly around 5 days of high intensity chemotherapy, and that's called the lymphodepletion. And that's a process basically we use that to deplete out all your existing immune system to make a space for the new one. And this process is really important because previous research has shown that without lymphodepletion, the efficacy can be reduced by more than half. And so then afterwards, the lifileucel or TILs will be infused into the patients.
Okay.
Yeah, I would say it's almost like replacing, partially replacing or reconstituting the patient's immune system with the T cells that are much better at recognizing, fighting off the cancers. It's kind of like re-engineering of each patient's immune system almost.
Wow. Yeah. So these billions of T cells flow through the person's body,
yeah,
and find the cancer cells.
Yeah, and actually we have research, that I did TIL decades ago, where we have shown that that in fact, that within these TIL cells, there are T cells that they can recognize, something called neoantigens on these tumor cells. So basically, neoantigens are basically the fragments of proteins that's displayed on the surface of tumor cells. From each patient's different unique mutations in their cancer.
So it's not even the cancer cells, it's fragments that were once on the cancer cells, and these new super T cells can find them.
Yeah.
Wow. So you've really just jump-started the body's immune system.
Yeah, re-engineering of the body's immune system.
Re-engineered. Yeah. Wow. So as you said, that it's still waiting FDA approval, could come soon by November.
Yeah.
What happens, knock on wood, that all the tests work and this is really effective? What does that now mean for treating people with metastatic melanomas?
Yeah, I think it adds a very powerful treatment options. So as you know, we know that from the standard immunotherapies like Keytruda or Opdivo or pembrolizumab and nivolumab. We still see like half of patients that fail to respond to the therapy. It's advancement over the past, over the old days where we're using chemotherapy where patients really don't benefit from those. It's an improvement, but there's still some unmet needs for these patients, when they don't respond to those therapies. And then there was a very interesting study that came out in December of last year. It was a study done in Netherlands where they look at patients that have failed, like Keytruda or pembrolizumab or Opdivo or nivolumab, and they randomized these patients to receive the TIL versus another all immunotherapy drug called Yervoy. It turns out that the patient's duration of response and response rate were much better in those that receive the TIL therapy. So I think that's going to be where I think the FDA approval may come. It's going to be targeted toward people, at least initially, it's going to be targeted toward those patients that have failed the standard immune checkpoint therapy.
Okay, so if they failed those other standard immunocheckpoint therapies, this is another option that will hopefully help a lot of people.
Yeah.
Okay, so again, once this is approved, and this is it's for metastatic melanomas, but this whole concept of taking out T cells, re-engineering, we've talked to other people, and CAR T cells is a similar thing, and even blood and bone marrow transplants are in a way the same thing. Yeah. But this concept of re-engineering the body's immune system and T cells, is that going to spread? It's already, but is it going to continue to spread to more and more types of cancer, and maybe even this drug or this TIL process could be used for other types of cancer?
Yes. I'm glad you asked this question. I think one of the excitement of TIL is that this can be applied to any cancers that have that are immunogenic, that have these really dense T cell infiltrates. And as a matter of fact, the company Iovance has been testing the TIL approach with other cancers such as head and neck cancers, triple negative breast cancers, lung cancers, and many other type of cancers. And I think so far some of the early studies have indicated potential efficacy in those settings. Again, the data is still preliminary, but there is some really encouraging signs that we're seeing. So again, this is I think this is a very exciting approach to the immunotherapies.
So I kind of want to finish up with a question. It seems to me that you and others like you at this stage in this, you're kind of pioneers in developing sort of a whole new, within immunotherapy, you're developing a whole new area of it that has tremendous possibilities. So what's that like to be in on and part of researching something that has this potential?
Yeah, I think we're all do this in the aim of improving patient outcomes in the future. Again, being an academic physician, I'm never satisfied with the status quo, right? Because there's always patients that I treat that fail to benefit from the standard FDA approved therapies right now, and I think it's important to create new options for those patients. Yeah, otherwise they just won't do well. And so this is an effort that kind of drives my professional efforts on a daily basis. Right?
So you've already treated patients through the TIL process that have done well.
Yes. Yeah, I remember a story of a I was in training at MD Anderson at the time where this TIL trials were just being developed at the time, and that was like 2009 or something like that. That was even before Keytruda and Opdivo were approved at the time. And I remember the story of this a young girl who's 17-year-old high school student, had stage 4 melanoma, and was given almost like a death sentence of very very poor chance of survival. And so this patient decided to participate in the TIL clinical trial that was going on at the time, and experienced a complete response.
Wow.
And she was able to graduate from high school, went to college, got married, and have kids, and was able to live a normal life. And I mean, just to see that kind of transformation is incredible. And I think that's what excites me about this, the potential of this kind of therapy.
All right, it's that kind of passion and excitement that leads to these breakthroughs.
Yeah.
Okay. Well, thank you for sharing this great information, and good luck as you continue down the TIL treatment line and helping more and more patients.
Yep.
This podcast is brought to you by the Ohio State University Comprehensive Cancer Center, Arthur G. James Cancer Hospital, and Richard J. Solove Research Institute. For more information, check out our website, cancer.osu.edu.